Our record is on, guys. So thank you. Welcome to our live here on Facebook. This one was recorded live during a live video or during a live stream Facebook under African Transcribers page if you want to follow. So guys, welcome to my class. Sanibonani, Dumelani, Avsheni, hello, I welcome you guys. So if you can hear me, you can comment there. If you can't hear me, guys, then we'll see what we'll do when time goes on. Guys. So okay, Life Science Grade 12 2024. During the day or yesterday, today is Tuesday, and on Monday, this is what we were doing on my previous YouTube video. This is what I was doing, a concept of inheritance. So guys, we talked about, I tried to explain many things. I tried to make you understand this concept of inheritance, genetics and inheritance. Guys, what do we start, or what do we, let me go to our caps, and from our caps, this was during our class, and after watching the class here, I saw that I did a mistake here. Gregor was supposed to be G-R-A-G. It's Gregor. You see, Gregor, O-R. Don't want to do mistakes, guys. O-R, because these are the words that they will ask you mostly. The father of genetics, Gregor Mendel, is a father of genetics. And then we tried to explain what is a chromatin. Let's go to our board from here, because this one is revision. We are just doing a revision. We are just doing a follow-up, what we understand up to so far from our previous class. So guys, from our previous class, what we can understand, the father of genetics, Mendel, Gregor Mendel, this is the guy, or he was the priest who helps us, or his findings helped us to understand the concept of genetics. We did say, what is genetics? We did say genetic is the study of how genes or traits are passed down from one generation to the next, ne? And then we talked about inheritance. What is inheritance? Inheritance is the action of inheriting. So while someone is inheriting something from someone, that process we call it inheritance. So I also realized that I did a lot of mistakes, guys, when I was referencing to capital letter A and small letter a. Somewhere I was busy saying capital letter B with small letter b, but I was writing something like here. I was writing a small letter a. Because guys, I was overwhelmed, as you can see, guys. This was my first class. Then also this one, it is also my first lesson. So we talked about what is gene. A gene is a portion of DNA. We talked about this, guys. If you missed it, go back to our previous video and study this. Then you come back here, and then we do this follow-up. And then from our follow-up, we can say that, let me use white, just a follow-up. After here, we are going to do that correction, and also I also gave you a activity there. So okay, we are going to say, in a zygote we have two chromosomes. In a zygote we have two chromosomes, one from the father and one from the mother. And then even here in the mother, before the meiosis, the gametes, even in the father, we had two chromosomes. And then from there, one chromosome from the mother. Let me do this. It's fine. Something like this. So we had two chromosomes from the father and two chromosomes from the mother. And then after meiosis, you learn about meiosis. After meiosis, one chromosome will move, or one chromosomes here will come, and then, okay, firstly meiosis, these two chromosomes will form four daughter chromosomes, cells, and then you still understand that in the mother, these three will degenerate, and only one it's going to form an ovum. And then this side, further, we are going to have also a four sperm cells. Then these sperm cells, one of these sperm cells is going to merge with this ovum here. So let's just take an example that this. Let's do like this. I want my example to be understandable, and then okay, it's fine. Here we'll have two cells like this. Also this side will have two cells like this. This one are the one that is going to degenerate. Ne? So let's just take for an example, one cell here, this one white one, the white one, it's going to merge with the red ovum here, and then they are going to form two, one zygote with two red, with one chromosome, one white chromosome, and one red chromosome. Ne? We understand this. And then when these two chromosomes, this one is a zygote, the zygote here there. So this zygote will bring it here. These chromosomes will come and aligned. And when they come together here, they will be shaped. On a human being, we know that there are 46 chromosomes. And then we are only going to use 2 for the referencing and breaking down of things. So they can also ask you on an exam, during an exam, why do you is this cell a human cell? Then the answer will be no, because here we only see 2 cells, and then here we only have 46. Okay, so it's fine. Let's move on. This is what we have learned up to so far, and then we learn that when these 2 chromosomes meet, or when these 2 chromosomes, when they are identical, they have the same shape, same number of genes, same what, they come and stay together, and then we call them, they are the homologous. You still remember this, and they are the homologous chromosome. Why are they homologous chromosome? They are homologous chromosome because of the same shape, first of all. Then the second thing is that at different locus, we talked about this, different locus, and they have the same gene. Another thing, at the different locus, they have the same gene. I said this one, it's a locus, a position, and then when they are many, we call them loci. You get me? And then what we have learned now, we are going to only concentrate on one locus, on one loci. One locus, Masori. We are going to only focus on one loci, one locus. And then in this locus, we have something, we're talking about something called an allele. We still remember it. And then we said this allele is an alternative form of gene. This is an alternative form of gene at a specific locus. So we can have this allele here, we can have this allele here, we can have this allele here, but when we have these alleles, we know that in this point we have allele that have a gene at a specific locus. We understand that. And then we also talked about these alleles are being represented by alphabet from A up to Z. And then we said this A, it can be A capital letter and A small letter. Since you are going to inherit 2 chromosomes from one from the father and one from the mother, it means that in this loci, your mother, let's just make an example. Like we are having an example where we have locis, or we have alleles from A up to Z. Let's say A, B, C, D, E, F, G, H, up until Z here at the top. So it means that at this loci, which will be Z, it's going to match homologous chromosome. It means that another chromosome having a same locus with the same what? With the same allele. It can either be capital letter A from the father, and the mother, it can either be capital letter A or small letter a. So it can also be from both parents. The father can bring in small letter a, and then the mother can bring in capital letter A or small letter a. So different is the same. So here in this topic, we tend to say or to reference the whole chromosome as in one allele. We will find that this chromosome, they are saying the chromosome, this one is going to have an allele of A, and this one chromosome is going to have an allele for capital letter A. That's the first combination, Anil. First combination, while we are using A up to Z. That's what I was trying to explain previously. And then another thing, another combination, we can have an allele where a father is giving you a capital letter A and the mother, she's giving you a small letter a. Or we can have a situation where a father is giving you a small letter a and the mother, she's also giving you a small letter a. You get this, right? So you'll find that we have a entire chromosome, and this entire chromosome is only represented by small letter a as an allele. Doesn't mean that that chromosome is only carrying that gene only, because we understand from our previous DNA replication that this chromosome is also made up of a chromatid, and this chromatid, it's a long chain of DNA. It's a long chain of DNA. In this long chain of DNA, we also have the histone here, the histone. We also have the histones here. But in this long chain of DNA, we have a portion that codes for a specific protein, and that portion we call it a gene. So you get me? So this chromosome can be classified as an allele of allele A, but that doesn't mean that that chromosome is only coding for that one gene. Also, due to our understanding and illustrating of things, we are only going to pick one allele for that one chromosome. So here one allele will represent one chromosome, although that won't be the case, because we'll have different location with different genes for specific characteristics. Let's say, for example, A, B, C, D up until Z, and then A is for allele, it's for gene for skin color, B for long nails, C hair color, so forth and so forth. So okay, we understand that, right? And then from there, we have alleles, we have gene, and then these alleles, we understand that these alleles are being presented by a capital letter A and small letter a. And in these two genes, we now have we can now differentiate them. I told you guys that this one now it's a zygote. This is a zygote. This one is a zygote with one chromosome from the father and one chromosome from the mother. And then when these two alleles are the same, they are going to form a zygote, or they are going to form a zygote with identical alleles. And those zygotes, we call them homo. You still remember it? Homozygous. And I told you, when you see these questions, make sure that you are aware that you are differentiating between homologous and homozygous. Because I also saw another question where they were like a zygote having identical alleles, and then the first choice was homologous, tripping you there to choose homologous instead of homozygous. And another difference, these two, because they're the same, okay, these two, AA and AA, they're the same, they will be homozygous. And these two, because they're different, they will be hetero. We talked about this, heterozygous. So if you have problem with this, you can go back and learn. Okay, it's fine there with homozygous and heterozygous. So we said this because this one is a zygote, and then in this zygote we are still referencing that zygote as in a genetically level. I'm not sure if I'm making mistake, if I'm saying this correctly. We are still referencing this zygote at a genetically level. We are still referencing it at the as the chromosomes. We have a chromosome that have allele A and A. If this allele A, it's going to code, or this gene, it's going to code for a color black or a color white. Ne? So if this allele, they are going to code for a color black or a color white. We have also, I talked about another term that they will use here. When we said a capital letter A, they will be dominant. You can also hear, guys, when you are dominating something, it means that you have power over something. And the one that is being, that the A is, it's the capital, the caps, they are having power over the small letters, and these small letters, we call them recessive. I talked about this, Akir. And then we also did we also went to our textbook and read and do a follow-up. So when you did what we are saying, now you know the capital letter A and small letter A. Then here under AA and AA, you understand now that this one is dominant, this one is dominant. It means that if capital letter A is for the color black, and then small letter A will be for the color white. And here, since color black is being represented by a capital letter, and this capital letter, we say is dominant over the small, it means that this person here is going to be a black when this person is born. When this zygote is born, this zygote is going to be black. And then here the second example, we have capital letter A, which is coding for the color black, and small letter A, which is coding for the small letter color white. And then we say, because the capital letter here A is dominant over the small letter A, it means that this capital letter is going to win. If you are saying these two, they are going to fight off which color is this baby going to be. The A, its caps, is going to be to win over the small letter A, and then the baby there will be black. And then the last, we have small letter A and small letter A. And this small letter A is recessive for the color white. And here there's no way that all of them they are losers, and they are recessive. That's what I used to claim them with. They are losers. So they are fighting amongst one another. The A will win, and then this person will come out as a white person. And then we also had another definition that these colors that we are coming with here, these colors that we are saying, if this win with this and this, these colors we are saying we are going to call them the phenotype. I talked about this. So these phenotypes, these are what we are going to say it's what we see on the outside. And then here, this one, they are going to be phenotype, and this AA, capital letter A, small letter A, capital letter A, small letter A, they will be genotype. Then genotype, gene level. This is the gene level. This A, it's representing this chromosome, and this chromosome, now we are talking about gene. From this chromosome, we'll have an allele, it will have a gene. Hence, when we want a genotype, we want these letters. When we want a phenotype, we want what is going to win when these two genes are combined. Are we together? Thank you. Then from there, you understand the most part. And then we also have Mendel as a father of Gregor Mendel as a father of genetics. The guy came up with three laws or principle. The first one, segregation. We said this one is separation. And when we separate two alleles from the father, when we separate two factors from the father, and then make sure that each and every cell got that factor. And then the second one is dominant. The dominant one is saying that dominance. Then the dominant one is saying that these two separated factors here, they come, it's either one of them can be dominant or one of them can be recessive. And if one of them is dominant, these genes that they are going to code for here, they are the one that we are going to see as our phenotype. Phenotype. Then if you can explain this dominant thing over recessive things, you won't have a problem explaining you won't have a problem explaining a principle or Mendelian principle of dominance. And then the third one, assorted. Ke tsela tsa hore during metaphase, the chromosomes are aligned at the equator randomly. They align at the equator randomly. So due to this, they are being separated, and they make sure that there's a variation. But this one assorted, they don't like bother us with it very much. On some books, you'll find that it only bring you segregation and dominance only, because this assorted, I heard that there's a, like, when you those chromosomes, they also have those chromosomes, because these chromosomes, they don't have one allele. Like these chromosomes, because they don't carry out one gene, it's not like one chromosome, one gene, because they carry out many genes. So it means that the genes can be now found on both. Like in this chromosome, we can find a locus for a brown skin, and then in this chromosome, we can also find a locus of a brown skin. Ne? So okay, it's fine. Thank you, guys. Let's go to our activity. So now you understand. Let's go to our activity. Let's do our activity, and then that's it. And then we'll keep this short and simple. And then with our activity number 1, it says traits, genes, and alleles. And then it will help you guys, it will help us guys to understand the question and understand what the question wants. In life science, when you deal with life science, they trick you in this life science. That's where you lose marks. That's where you find that most people, they don't get 100% out of 100% with life science. They can get 100% with physical science, they can get 100% with mathematics, but when it comes to life science, because there's some tricking other things. And then okay, it says the paired letters in the diagram below represent alleles. Let me use a red color. Thank you very much. The paired letters in the diagram below represent alleles of a gene. But a number of genes for different characteristics are shown. Write down the relevant letter A up to D for, but number A, the homologous dominant state. Homologous, we want to see the same. We want to see those who are the same. Which one are the same? We have B. It's the same. B. We are still, we haven't given an answer. We are still analyzing our question. Homo means same. We have SS. We have SS. And then we only have these two that are the same. But they are saying, write the one that is homologous, same, but dominant state. It means that we have same, but both capital letters. So we go and say both with one, the one with having both capital letters is this number B. So the answer here will be B. So you saw it means homologous, same. And then same, dominant means capital letters, and then that's how we found number B. So you see, sometimes you might be not understanding what is homologous. You might be maybe not even being able to explain to someone what is homologous dominant state. But if you can put it down and write homologous, show me the one that is homologous, you know. Homologous, they are the same. Then after that we choose homologous recessive or homologous dominant. Now if it's homologous dominant, it means that two capital letters. So number B, two alleles from different genes. Two alleles from different genes. So here we want we have two alleles but different genes. Then we have heterozygous here. Then we'll have a capital letter. With our example, we'll have a capital letter and a small letter. Which one of those? We have this number T, number TT, number RR, and number two alleles from different genes. These are two alleles from different genes, and we also have a number two alleles. No. They want two alleles from different genes. Like they want two alleles of the two alleles here. Here's two alleles from different. So you see, even D here, it also gives two different alleles. But the problem is that here they want the two alleles, which is mean that this one will be A. This one will be A. These two, we have two alleles from different genes. Ne? We have two alleles from different genes. It means that we have this gene and we have this gene in this A. And then number C, the homozygous recessive state. Homozygous, same, but recessive. Means that we have small capital letters here. Small capital letters. Which one do we have? Which one of these do we have as small small letters? Number C. They are the same, yes. Are they recessive? Are they small? Yes. So here, number C. And then number D, the heterozygous state. Here's what was causing me a confusion, because I also have this question and also have this question. So here it says the heterozygous state. The heterozygous state is number A and number D. So we'll say D and A, because here they only want heterozygous state. Heterozygous state, those that are not the same. Are these S and S the same? No. S and S are sorry, I wanted to do the reverse. Are S and S the same? Yes. No heterozygous. S and S, yes. No heterozygous. Capital letter B, small letter b, heterozygous. Capital letter R, small letter r, heterozygous. Different. Capital letter T, small letter t, heterozygous. So now from here, you'll have to understand, you should understand by this time that if I can come and say, give me a zygote, or give me two alleles that represent a homologous dominant, then you know homologous, same, dominant, capital letters, then you are going to give me something like this. Then if I said heterozygous, one is dominant, one is recessive. If I said homozygous recessive, I mean those that are the same but small letters. We are going to use this information when we go on, guys. Okay, let's move on. We don't have time. And what is the relationship between gene and protein? What is the relationship between a gene and a protein? Gene is a portion of DNA that code for a protein, right? Or a protein is being encoded from a messenger RNA, that messenger RNA is from the gene. And however you want to explain it, goes 2 marks, guys. So if you can say that a gene code for a particular protein, you can get your 2, because that's the relationship. Or proteins comes from the gene. Or the gene produce mRNA. However you want to do it, this mRNA go to and encode or produce a protein. So here's the relationship, guys. You understand the relationship. So I'm just picking up where maybe you want to play with words. So guys, let's move on. What is an allele? Here's the question. But what is an allele? King allele. An allele is an alternative form of gene. Alternative form of gene in a particular or specific locus. This is an allele, and you did it there. You can go back and see if this is correct or not. But the allele is an alternative form of gene. Why do we say an alternative form of gene? Because this allele doesn't say because it's an allele, there's a specific gene that we can find at an allele. There's no specific gene you can find at an allele. You can find gene for a hair color. You can find gene for a eye color. You can find gene for long long ears, tongue, whatever. So this an allele is an alternative form of gene. So we can have different gene at a allele. So one allele doesn't mean that that allele is specifically for that that gene. Then number 4, what terms describe a pair of alleles that are the same? Homozygous. Homozygous. And then different, heterozygous. And then write a definition of a homologous chromosomes using the terms gene and the allele. So here you will have to say that homologous chromosome are chromosomes from the father and the mother. This one, they have the same gene. I create the same using the terms gene. They have the same gene and same, or the alleles can be different, although the alleles might be different. They have the same gene, and then, how can we put this? They have the same gene, and although the alleles may differ, they have the same gene, although the alleles may differ. So these homologous chromosomes, they might have the same gene, and although the alleles may differ. So yes, these chromosomes, they can have the same gene, let's just say at a same locus here, or they may have an allele for a specific gene. You can say they can have alleles for a particular gene, at a specific locus. So it means that if this is a chromosome and this is a chromosome, it means that in this locus, same locus, specific locus, or specific or same locus, we will have, if here we are going to have a gene for eye color, also here we are going to have a gene for an eye color. That's how you can see these are homologous in terms of genes and allele. And how are alleles represented? They are represented by alphabets from A up until Z. But fill in the table below with the missing genotype, phenotype, dominant or recessive. Then during my class I told you guys that this genotype here, this one is just a definition of our genotype. Let's do this since we don't have time. It's saying homologous dominant, the phenotype and alleles. Here we can say that we are dealing with short. If it's not short, it's tall, like it. Then if it's not there, we have a capital letter and a small letter for a short. This one for the tall. And then they're saying homozygous dominant. They are the same, yes. T, homozygous dominant, T. And the phenotype will be tall. The capital letter one will win either way. And then here they are saying short, TT. And then we come here, are these the same? Yes. It means here we'll have homozygous for this same again. Homozygous, are these capital letters, small letters? These are small letters, will be homozygous recessive. And then the last one, they leave it like this. And then it means here you will have to work it out again. Alleles, they can be like this and be like this. It means here we shorter this and this. And then phenotype, if these are like this, do you understand? Like they want you to tell them if you understand this. If they're like this, is this going to be tall or short? This one is going to be tall because there's capital letter here, this capital letter is going to win over the recessive, right? And then the phenotype here, the phenotype will be tall. And then the genotype, how are we going to express this? This one are the heterozygous. You can write one dominant, one recessive. Nothing much is going to change it. Okay, it's fine. And then number 8, draw a pair of homologous chromosomes. You're going to draw a pair of homologous chromosomes. We can draw that pair of homologous chromosomes. By this time you understand that this is a chromosome. Also guys, we also have people who are still was still confused by these drawings of mine. Some of them, they say, hey, what if I was told that this is a chromosome. This is a chromosome, guys, after interphase. And this is the chromosome after interphase, after DNA replication. After DNA replication, we are going to see this one and that one side. When we see this, we know that the cell now it's preparing to undergo meiosis or mitosis. But when they are not undergoing meiosis or mitosis, the chromosomes stay like this. But draw a pair of homologous chromosomes. You draw it, and then in life science, when they say draw, you also have to provide the diagram. Okay, you can just come here and say homologous. You are not wrong. Chromosomes. Here you also have two. You have one for the cell, you have one for the name. Before you can even go somewhere, it's out of 5, and these two, you get them. Homologous chromosome, we want to see 2 chromosomes, and then we also have to name here. Don't forget to name your diagram. This one's a name of diagram. And then Barry, label the chromosome with two sets of genes. Label the chromosome with 2 sets of genes, but one with homozygous dominant alleles. Then you are going to, here they didn't give you, same as previous, they didn't give you what do we call, they didn't give you alphabets, and then it means when now you are going to choose from A up until Z. And then they say one with homozygous, they are the same, dominant. It means here we want capital letters. Let's use capital letter B. We are going to have B and B, and then you do what, you label this, and you said homozygous dominant and dominant. And then from BB you go and say, and then another one, they say one with homozygous recessive alleles. We want small letters, and these are the same but small letters, recessive. And then you say this one, excuse, you'll say this one is homozygous recessive, ne? And then and one with, here they said 2 sets, here they said 2 sets of genes, but now they are giving us 3. And the last one, they said one with heterozygous alleles. It means heterozygous alleles, one will be capital letter and one will be small letter. And then you'll also label these 2 as heterozygous alleles, or heterozygous, one dominant, one. And then it's 5 marks, 1, 2, 3, 4, 5. And then that's it. I think doing this you'll be fine, and then 25 marks it will be yours. Then from there, next week we'll start here with Mendel. I'll start with tomorrow I'll start with Mendel, and then after Mendel we'll come and do the genetic crossings, monohybrid. So thank you guys, thank you for your time. That was it from me. I'll try, guys, and not waste your time by all means. So thank you guys, see you next time. Sharp. Peace and peace. That's it. Now we go to Facebook. As I can see on Facebook, guys, we are still the thing hasn't been, the thing is still typing there, so we'll wait for it to finish, and then I will cut off the videos. I'm not sure if I cut